we start today's workshop, I would like to introduce the people who are behind organization of this workshop. So the workshop is organized by the Career Development Committee of GSA. And although today Molly and me are moderating the workshop, but every individual member, as shown over here, has been instrumental in making this event happen. A special thanks goes to Erin and Jessica from engagement team for a lot of support with administrative and technical work. Uh, so before we start, another thing to mention is that there are certain ground rules applies to this workshop and an event like any other GSA event. So uh, this workshop follows the guidelines and code of conduct of GSA, and those code of conducts are there pasted in the comment box. So please go through them and follow those guidelines and be respectful to everyone. And other things to mention is that the views and opinions shared by the panelists today in the workshop is of their own and not of the organization they represent. And naming any brand or company is not an endorsement. So now I will just give you a short outline of how we will progress through the workshop today. So we will first go through the introduction of our panelists. As you can see, we have three great panelists from different pharma companies who moved from academia to industry. And so after introduction, we'll move to the main question answer session, where we have a set of questions, which is selected from the list that we got during registration process. So all of the participants have provided us with the questions, and we selected the kind of most popular questions. And then after that, we last 10 minutes, we will have a live question answer. So if you have any new questions, you can put it in the chat box, and we will go through them at the end and try to get the answer from the panelists. In case of time restriction, if we are not able to answer any question, please do not worry, because we'll be compiling a resource package, and we will try to include the answer of the questions which are unanswered today. So with that, I think we can start our workshop, and I request Dr. Deepa Balasubramaniam to introduce herself.
Hi, everybody. Good morning. My name is Deepa Balasubramaniam, and I am currently a senior research scientist at Eli Lilly and Company. And I got my PhD at Purdue in 2011, and then spent a few years as an academic postdoc, followed by an industry postdoc, after which I transitioned into this position. And I'm happy to answer questions with my, that align with my experiences.
Thank you, Deepa.
I also work, sorry, I should have added this for some of you might know this. I also work extensively with this organization called STEM Peers in helping international students or people transition and also on visa related queries. So that's something I'm comfortable talking about.
Thank you, Deepa. We'll move to Dr. Timothy Hammond.
Hi, yeah, I'm Tim Hammond. I am a principal scientist and lab head in the neuroscience research unit at Sanofi in the Boston area. And I started my career getting my PhD at George Washington University in Washington, DC, and then went to go do my postdoc at Boston Children's Hospital with Beth Stevens. And then from there transitioned to this position about 2 years ago at Sanofi.
Thank you. We finally have Dr. Shivabrata Sanyal from Calico.
Thanks, Saurabh. Hi, everyone. Great to meet everyone. This is very, very interesting. Of course, I should say that I never thought that I would be in a panel like this, but it's frightfully interesting. So I'm Sanyal. Very nice to meet everyone. I am head of in vivo sciences, which is basically preclinical pharmacology at Calico. I've been at Calico for about 5 years. Prior to that, I was at Biogen for about 4 to 5 years. prior to that, faculty at Emory University for another 5 or 6 years. So I've had a sort of a long road. It's been an interesting ride, and hopefully we can share some of that with you if it's helpful at all.
Thank you. So now we can move to our question answer session, where we will go through the set of questions that we collected from the participants. So the first question is, why did you decide to choose a career in biotech? And how did you know if this was the right fit for you? The question I would go to Tim.
Sure, I can kick it off. And, I think there's sort of a number of different reasons why I ended up in pharma. And I think from a sort of scientific interest perspective, much of my motivation and drive in science has been, understanding mechanisms to help cure disease. And that's really sort of been a theme in most of my work during my career. And, I started off, sort of studying, basic mechanisms, but then quickly sort of found that, I was very interested in sort of the translational aspect as well. I think from a more sort of practical perspective, I think for a long time, it was always the plan to, start my own lab in academia. And that was sort of how I structured my career. And, even got to the point where I was interviewing for positions in academia. And I think being in the Boston area, I was sort of exposed to other opportunities and sort of the degree and breadth of basic scientific research and discovery that goes on in pharma and industry that I wasn't really aware of earlier in my career. And, sort of got excited by this idea that I could run a lab in industry, still, sort of work on topics that I was excited about and interested about, but also have the potential to take those ideas all the way through to the clinic.
Thank you. So our next question is, what area of biotech or pharma were appealing to you, and how did you narrow down on your search? I would go to Sanyal for the answer.
Sure. I mean, I guess when I started out, long story short, I think I was interested, oddly enough, in plant biotech. Why? Don't ask me why, I just was. But then as I sort of moved through the academic ladder and got into neuroscience, once I got into neuroscience, I just got hooked and there was no looking back. I'm interested in various aspects of neuroscience. And then, focusing in on a particular area, I've actually moved around in neuroscience a fair amount. So, all the way from thinking about plasticity mechanisms in the brain to right now more focused in neurodegeneration, but also because Calico is such an open-ended company, really thinking about the impact of age on the brain and on the nervous system. So it was really driven by interest. I should say that some of this, as all of us will recognize, sort of self-fulfilling. The more you do, the more interested you get. And then when you start making contributions, then it drives you even more. So then the fit sort of merges with your experience and your knowledge in that area. So that's basically why I'm in the field I'm in.
Thank you. So next we'll move to Deepa, and the question is, how did you apply to the position, and did you adopt any strategies or use any specific resources that help make the process smooth?
So this is multi-pronged for most of us, right? So I did apply for positions as I found them on the web. I did do a few of them, but I was very focused in the positions I applied for. I did not apply to every position I found because I kind of knew what I wanted to do or what my strengths were, in the sense that I wanted to be in discovery sciences. So that's where I ended up. I'm in the large molecule discovery group at Eli Lilly right now. The one thing that terribly helped me was networking and getting to know people, because almost every opportunity I've ended up having in hand was because I knew something about the company or somebody in the company who could either tell me more or guide me through some things. And the biggest winner for me, in some ways, I was also lucky that during my postdoc, I was very open to my PI about what kind of opportunities I wanted. I wanted to learn as much as I could and figure out where I wanted to end up, industry, academia. So I wrote fellowship grants on one side, right? I learned how to grant write, how to resubmit, how to fight through that. On the other side, I also took up every little industry collaboration or anything that we could find through the lab. And I said yes to everything. Yes, it was more work. It's, a few years of hard work and, putting my efforts and money, different pockets. But in the end, all that made me help choose what I wanted to do with clarity, and also built me a network that I'm ever thankful for. And the job I have today is because I worked with these people when I was a postdoc, and my transition was relatively easy.
So like networking helped you a lot in getting your current position.
Yes.
Right. So I'd also like to thank you, Deepa. I would also like to mention that if like I'm going randomly to some panelists, but if anyone else has anything to add, please feel free to jump in. Okay, so the next question we have is, did you require any additional training to better prepare you for the application and interview process? We'll go to Tim for the answer.
I think I'll second, the comments on sort of networking and having connections and companies. And, I think, really one of the main goals is getting your resume in front of the right eyes and, finding the person to email, or even if you don't know them particularly well, I think can go a long way. So, I mean, I think, in terms of sort of interviewing and preparing, we also had workshops like this at Boston Children's that I think were helpful. And, I think one thing that could have been helpful was sort of structuring your resume in the right way and sort of emphasizing the right things. I think the structure of an industry resume is sometimes a little bit different than an academic one. And, it's maybe less about sort of listing sort of every presentation and every sort of, and more so about really sort of like outlining your career goals, what you've done, where you're going, what your interests are, and your experience sort of first and foremost. So I think, having your resume set up the right way is important. And then, from the interview perspective, the other panelists might have had sort of different style interviews, but mine was actually somewhat academic style. I gave a presentation on my work, fielded questions from, other members of the research group here at Sanofi, and then, had one-on-ones for the rest of the day. So I, really sort of leaned on sort of my academic training in terms of giving clear and compelling presentations. And, but beyond that, I didn't have, much else in terms of training.
Thank you, Tim. So just to continue with what you said, like, I think we have many questions. Many people want to know the interview process. So is it like always you give a presentation like you do in academic settings and then talk to people? That's where it goes?
That's how we do them here.
Okay.
Yeah.
Thank you.
I can add a tiny bit to that, because just like Sanofi, I've interviewed, obviously, I've interviewed at a handful of mid-sized pharmas and big pharmas over time, and almost every one of them were like what Tim described, starting off my day with a 45-minute presentation, fielding questions, meeting with the team and other related groups that you would be working with. And then obviously there was a component of the HR throughout it. Since I do help people who go through this process, I think with the virtual environment, it's become slightly different in the sense that we don't have day-long ones anymore. They're more concise, and you might meet a fewer number of people only because of the way. But I think once this passes and we're back to the new normal, it'll go back to what it was. but this is very typical of a bench science or a research scientist PhD level interview in almost most pharma companies.
So just to continue, like during the presentation, you are looking at the scientific capability of the person, but then when they are meeting to one-to-one to other employees, so what are the qualities you try to get from that person? Like, is there anything that is important when you are interacting with other people to?
I'll feel this first, because I'll tell you one main thing that we've repeatedly discussed in interview groups is, how do you think that person will get along with the team? Right. So I've heard people joke that at the end of there's obviously scientific questions that are asked during these, when you meet with scientists, and some behavioral questions about how you managed conflicts or other things. But one of the biggest things that I've seen is, how do we think this person will get along with the rest of the team? Because I think that vibe plays a big role. Because at least where I've worked, teamwork is the biggest thing, because you don't get to do everything yourself. It's different from being a postdoc, or in what you get in most academic situations. Everybody does a little bit to get the greater good kind of thing. So being a part of the team is very important.
Thank you, Deepa. So our next question is, what challenges did you face during the transition process, and how did you overcome them? I'll go to Sanyal for the answer.
Sure. I think so. Yeah, there are obvious challenges. It's interesting. I've obviously went through grad school, postdoc, but I had my own lab for a while, academic lab, and the manner of working in an academic lab is it's different. And I'll go back to what Deepa just mentioned about teamwork. We all talk about teamwork, but you really have to be in industry to realize really what it means. And so one of the challenges is sort of a philosophical challenge. And a lot of people who've talked to me about this transition have said, well, I'm running my own lab, I want to go into industry, but I've heard that you don't have any freedom. This is the usual refrain. And it's a loaded question, and there's some truth to that, and there's some untruth to that. And so one of the biggest challenges is really genuinely learning how to work in a team, and genuinely learning how to align your interests, or seek out areas where your interests are aligned, and how can you contribute to the larger mission. It doesn't mean untold slavery. That's not the case. There is enough room for innovation, and especially those of us who are on the discovery side of the pipeline, there's actually a lot of room for innovation. But you do have to understand that it's not like getting a grant and doing your own work independently, and that you'll submit a report to the NIH and have some manuscripts, and in between, nobody asks for anything. It's not like that. And so therefore, the degree of justification that you have to provide, the consultations, the input and the feedback from teams and all of that, that's an ongoing process, starts on day one and just keeps going. So you get used to that, and that may be a little bit of a mental shift. The other one, has to do with how industry works and what industry focuses on. When you're doing academic research, oftentimes one of your goals, practical goals, is writing a manuscript, disseminating knowledge, creating new knowledge, and all of that is fine. In industry, it's a little bit different in that, sure, manuscripts and so on and so forth are fine, but there are these overarching goals, and it takes a while for you to understand what those goals are and align yourself. So when you're just making that transition, there's a period of just listening and learning. And you just have to do that, and you just have to learn, and there's a lot to learn. And when you do that, I think you slowly start understanding what really matters in that context. So it's a fuzzy answer, because the problem is a little bit fuzzy. It's just the way it is.
Yeah, thank you. So just to continue, like you were talking about teamwork, but whoever is working in a lab in academic settings, they are also kind of used to a teamwork, right? Is it different in industry in a way?
Yeah, so I would say there is a lot of difference in what there's a lot of flexibility in what goes as or gets defined as teamwork in academia, right? And so, yes, it's a team, it's a lab, and there's the overall purpose to the lab and so on and so forth. But by and large, I would say that people have their own independent projects, and there are collaborations, of course. But even collaborations work differently. You do your own bit, somebody else does their own bit, and so on. Drug discovery is such a complicated process. There are so many nuts and bolts that fit into this, especially as you sort of move along. And so there are two kinds of things, right? So one is everybody understands that for any kind of molecule to move forward, you need to do tox studies. So oftentimes you'll find that, and I'm not trying to disparage toxicologists by any means, but I'm just saying that it's the nature of the work. There you may find that somebody who's very, very good at what they do, things come in, they do their thing, they run a 5-day rat tox, and they give you the results and they interpret it for you. You have some questions and follow-ups, and that's fine. And then there is the other role, which is a more sort of strategic. How does something fit into it? How does something fit into the pipeline? What do you need to generate? What is the whole pathway? What is the lifetime or life cycle of a drug? There, teamwork can involve a large portion of the company. It's not just the next 5 people. It's not just your team. It can involve a large portion of the company. I'll give you an example. So for example, at Calico, we are very much a discovery unit, and AbbVie are our drug development partners. So much of the heavy lifting in terms of drug development, medchem, tox, ADME, all of that gets done at AbbVie. So our team is an extended team, but we need to work with them to try and forecast what's going to happen, what the next step is. There's all sorts of prioritizations involved. We have 10 different things going. All of them can't go together. 7 will work, or 7 will stop, 3 will move forward. Teams need to be aligned. You need to be very flexible, you may be doing something and you may never get to complete it because something else is of more urgent priority. So you move there. And the entire team has to align and realign. You can't just wander off and say, I'm going to just do this by myself and because I just like to do this. It's not going to happen. So teamwork there means really focusing in on the overarching goal. That could be your team goal, that could be your organizational goal, whatever that is. And you need to be flexible. Rarely happens in academia.
Yeah, thank you, Sanal. I totally understand that. Like in academia, we have, every individual has a specific goal of publication, which is probably not there in industry. And that's kind of.
I mean, you do publish. It's not that you don't do that whatsoever. You do that. It's not, I mean, that just is an example to say that, the practical goals are slightly different.
Right. Thank you. So the next question we have is, what skills are necessary for a research position in biotech, in addition to the skills that already we acquired through academic training? And I would go to Deepa for the answer.
I don't think you need very clear, I don't think you need anything extra, to be honest. The most important thing, in my opinion, is taking the skills that you've learned and presenting that in such a way to show how you could apply that or use it, right? And one of the things, this is a pet peeve or a personal belief, that as a PhD, you have training to learn any technique, any skill. You've gained your currency to do that, right? You've proved yourself again and again, especially if you have a postdoc too after PhD. So I don't think you are hired basically as a pair of hands, right? You're hired because you can think through a problem, you can problem solve, you can contribute to a team, you can maybe bring in an outside idea that the team's not already possessed. Now, I do have a role in which I have one main deliverable, or I'm kind of a part of a core team. I touch every project and I'm expected to deliver on it, right? But that doesn't define me, or that is not the only thing I do. So personally, we could talk about this, I think, in different aspects, but I don't see that you need a ton of other skills. But the most important thing is to distill what you have and present it in such a way where, bring it to whatever interests you, right? You could be working in a model organism in your PhD, but that doesn't mean that's not transferable, or what you've learned is not useful to apply to a drug discovery project. It's about how you think about it.
Right. Thank you, Deepa. Tim, do you want to add something?
Yeah, no, I think those are good, all good points. And I think, a lot of what you do in the context of the teamwork that we've been talking about is really, delivering sort of clear data and conclusions to help make decisions to help push programs and projects forward. And, I think like Deepa was saying, you need to be able to sort of distill down your work in a clear way and present it to many different groups that have many different scientific backgrounds, whether it's in talks groups or DMPK groups, or, people have a very wide scientific expertise as part of this broader effort in pharma and biotech. And I think, as someone with a PhD and someone who will hopefully lead a program, once you get here, you really need to be able to show that you can do that in a clear way, in a productive way. And I think tied to that is, also showing that you can sort of see projects through to the finish. And there are many different ways that could be evaluated, whether it's through publication or just sort of a clear arc of the science during the presentation, or problem solving ability, this type of thing. I think those are all sort of important things that we look for.
Thank you, Tim, for the answer. Next, we have the question is it challenging to move to a pharma scientist position from basic science background? I think Deepa already touched upon that. Do you want to add anything more, Deepa?
I don't think I have anything. I don't think so, because I'll use myself as a classic example. I'm a chemical engineering bachelor's with a biochemical engineering master's with a structural biology or more biochemistry based PhD. And I work in large molecule discovery. I do study protein-protein interactions here. That's my, bread and butter. But if you translated every experience I've had, it's not, I didn't do the same thing again and again and end up where I am. And I hope I don't stop here, because one of the things that completely thrills me about being in pharma is the breadth of what we do. So I hope that answers people's questions, that no, you don't need, you can turn around, or you can flip what you do, you can use it. You have the skills. You just need to broaden your, think outside the box, or take a deep breath and see what you've learned and how to apply them.
Thank you, Deepa. So many people from actually Drosophila Genetics have asked this question, and I'd go to Sandel because I know he has that background. Do you want to add anything, like someone from Drosophila Genetics want to move to industry? Is it challenging?
I'll go back to what Deepa said. I think I'll preface it by saying that the skills you learn, and this was mentioned even earlier, the skills you learn are the skills you learn. And so if you can think through things, if you can think through problems, Drosophila Genetics is a fantastic place to actually learn how to do that. So, so that's actually a plus point. Now, the thing is that it's also true that for some, not all roles are made the same way. And for some roles, if somebody is looking to add to their mouse physiology core, and you apply with the Drosophila Genetics background, and that person wants you to start tomorrow, it's probably not a good fit. That's okay. But there are plenty of other roles where your problem-solving skills are what are of interest. If somebody wants to start a target discovery initiative in a rare disease, and Drosophila Genetics is what you've done, but you're open to thinking about cell culture or whatever the methodology might be, then actually they'll look at how you think, how you plan your experiments, how deeply are you capable of going into the literature and separating out the important bits from the fluffy bits. So there is some degree of limitation when you specialize in a model system that is not immediately used in an industry environment. But I would say that those roles you can easily identify, and there are many others where your skills as a scientist are what are valued. It's a small anecdote. Early on in my career, I happened to meet someone who is also a Drosophila geneticist, and very well known. And he told me that, look, Sanyal, the thing is that I don't understand the first thing about music, but if I'm in a room and there are 5 musicians who come into interview, I can probably tell you who's a good musician, because you can identify those skills. Even as he was talking, obviously, as an anecdote. But I think I've seen that repeatedly in my career progression, but also in many of the folks that I've hired. Oftentimes you'll interview and you have their CV and so on. And I actually tend to pretty much interview. I look at all the CVs myself, so it doesn't really go through HR, it goes through me. I absolutely make sure this is the reason why I make sure that I look at everything. I don't want those keywords to eliminate people. And then when I interview, I really look for what I would call the raw material. How do you think? Who are you? What have you done? And it doesn't really matter if it's a role where I don't want you to be able to do one particular thing from day one. Usually the people can be trained, and the good ones, they just pick things up. It's fine.
Thank
Yeah,
you,
I think-
Sanyal. Yeah, sure.
I think maybe on that side, I completely agree. And I think the only thing you have to be willing is to be sort of more flexible and inquisitive once you get over. I mean, if you expect to only work on Drosophila genetics moving forward, you're probably going to have a hard time. But I think if you're, as Sanyal said, just a scientist who's interested in the questions, interested in the biology, the application to disease, the questions are the key aspect, and the methods that you use to get there are, maybe not, there are different tools obviously that we use within our groups. Sometimes it's more in vivo focused, sometimes it's more in vitro, but I think everyone sort of wears a lot of hats to sort of answer the questions that we need to answer. So
Thank you, Tim.
I want to add one quick thing because this came up. So even if you end up in a position that requires one key skill or two key skills, right, that doesn't limit you. And the reason I'm bringing this up is I do that. I was hired here in this position because I had hydrogen deuterium exchange mass spec expertise. And I also got my industry postdoc for the same reason. I've worked in different small molecule, membrane protein, large molecule, like I've done a little bit of different things in different places. But that hasn't limited me in being involved in projects or, using my expertise outside of what, I know other things and I'm interested in science as a whole. So I've had, you can make opportunities to be parts of projects, be part, understand biology, understand mechanism of action. So I've kind of learned how to navigate that too. So again, even if you end up in a position where it seems like you're being hired for those 4 skills that you have, and the company needs it now, you do deliver on those, but that doesn't limit you. So don't bucket yourself, or don't worry that, oh, if I end up in a position like this, it'll like put me into that depth or into that one silo and I'm stuck there. It doesn't. You still learn a ton of other stuff.
That's actually a very good point. I think it's a lot easier, I would say, to shift laterally within, especially these large companies, because there's just so much going on. It's an entire universe in these companies, and it's much easier to do that than even in large universities where you have to go to a different lab or to a different department or something. It's actually, if you look for it, people are more than happy to help you, and they're willing to accept your help. It's much easier to do that, and you really should do that. Absolutely, good point.
Thank you, Sanyal. So the next question we have is, for an international student or postdoc working in FJ or whatever visa, how difficult it is to get a sponsorship for a scientist position? I would go to Sanyal.
I would actually ask Deepa to address that, given that you have a background in advising others.
Sure. So I wouldn't say it's easy, but my points have always, that doesn't limit you again. Okay. I've won the war more than once on this, and I know of many who have. So I'm happy to, talk to people individually if they need to get specific advice, because there's different categories. Each one of those F, J, and H have different reasons and different things to learn about, to learn to transition. It's doable. Many have. So you have to learn things. You have to know about things. This is another piece I tell people. You have a PhD, and so you're very, very capable of learning new things and acquiring knowledge. Learn the immigration laws, learn them in and out. It's just part of your job search and your career planning. It's not it's not for somebody else to figure it out, because sometimes each one of these cases become an individual path to navigate. Unfortunately, for each of us, it's different. And it's not impossible. For anybody who's international in this panel and wants to hear, I've been in this country 20 years of this year. I only got my green card last year. I've been on every visa category. And as I've said, I've worked for industry for the last 7 years. So it's doable. Don't panic, just learn. And I think the number of resources to learn about this have also expanded immensely over the last few years. It wasn't the same when I ended up in the US. 2001, right before 9/11, was not the best time to start in this path, but we've come a long way. So again, it's not the limiting factor. Don't worry about it. It's just one more factor that you need to add in. And yes, you will probably have a few more rejects or a few more we can do a visa, but don't let that stop you from trying if this is what you want.
Thank you, Deepa. So the next question we have is, what does your day-to-day job look like? How is it different from academia? Deepa, can you answer again?
My day-to-day, I do some lab work, as in I plan whichever projects and whatever experiments are there. And I also have somebody who reports into me who does the bulk of that work. And I'm thankful, and actually it doesn't work like this for everybody, but that's how my setup is for various reasons. I have meetings to go to, meetings that are project-oriented, meetings that are, like Sanil was mentioning, our group, our department, overall goals and overall, different pieces of the puzzle. So it depends on a given day. And then there are days when I have a few hours of, I've blocked my calendar off in hiding because I have to analyze data. The kind of work I do, there's more analysis involved than, and I've learned over time to actually put that down in my calendar and preciously, keep the time, because if I don't, I end up in meetings, I end up in other things, and then I still have my work to do. So it's a mix. It's a mix of meetings for different goals or projects. There's lab work involved, of course. There's training other people involved, and a bunch of data analysis. And of course, as we talked about early on, doing all that analysis and putting it together in a clear, concise manner and delivering it to the teams so that they can make the decision and move on. So there's that too. And of course, just like in any other scientist world, there's fighting your instruments, fixing them, troubleshooting stuff too, which is actually fun, in my opinion.
Yeah, thank you, Deepa. So if I ask you, so you said mostly either research work or meeting, does it also involve managing people? Like, is there people?
Case, yes. In my case, yes. Probably more in Sanil's and Tim's, because they have bigger, Sanil probably has a bigger group being a director, and it's probably more of a management role. But as a scientist, yes, I do manage other people, but and I also kind of mentor and help people who don't really report to me, because either the science aligns or we align as some people, you just have that kind of relationship, and growing other people is part of your job.
Awesome. Thank you, Deepa. So the next question we have is, and it's a popular question, what does your work-life balance look like in your position? We'll go to Tim.
It's actually, I can't complain. I think we're certainly very busy, and we have sort of a few programs that, need varying, bursts of attention, beyond normal at sometimes. But, I would think generally speaking, most people who work here in sort of the discovery part at Sanofi, are not working weekends unless absolutely necessary for sort of animal dosing and for iPSCs or sort of maintenance type work, at least on the scientist side. I think, most people work sort of an 8-hour day. So I would say the work-life balance is probably better than in academia, to be honest. I think, when you need to work hard, there's an expectation that you do. And sometimes that, if there's an important presentation or if there are important experiments, then, there might be sort of, outside normal working hours. But I think typically it's sort of an 8 to 5 limited weekend type schedule. So I would say in pharma, at least, it's quite balanced is what I've found.
Thank you, Tim. Do other panelists also agree, like it's better than academia?
I'd have to agree with you. I think it absolutely is. The only thing that I will add is that it depends on the company a little bit. I mean, if you're in a smaller startup, I can imagine that the pace of that, or at least the perceived pace of that, and the pressures of being there all the time, are going to be different from a larger company where roles are much better defined and things are more organized. So that is just true. But in general, I would say in companies, I think the expectation that we will have a work-life balance at least is much higher than in academia.
Thank you, Sanyal.
And I think just in the last year and a half with COVID and everything that's happened, most companies are being more flexible, being more open, and that, I think there's a clear, I think there's a better understanding of what employees need to be supported to deliver. So I think it's only going to get better as far as work-life balance in industry goes, in my opinion. Academics wise, I'm one of those anomalies who had two very supportive mentors in the later stages of my academic career. So I didn't have a horrible academic experience later at the second stage of my life. So I have.
I think there is some issues with connection. Yeah, maybe we can continue because I think there is some connection issues with Deepa.
One point that I think was sort of touched on is defining expectations for positions and deliverables, which I think in academia can be very sort of nebulous. Get a paper in 4 years, or I think sometimes those aren't very clearly defined. But I think in my experience in industry is that the roles are much more clearly defined. The expectations are as well. You're having mid-year sort of evaluations by people who manage you. You give evaluations, and I think it's always very sort of clear where you stand and if you need to sort of change the way that you're working, or so I think that sort of factors into the balance as well as sort of understanding what needs to be done and if you're achieving those goals or not.
Thank you, Tim. I think Deepa has some issues with connection, but probably she will join back again. But we'll continue in the meantime. So the next question is, what do you love the most about your job, and what aspect do you dislike? To Sanyal.
Sure. Yes, nothing is completely perfect, obviously. I think what I like about the job, it sounds very cliched, but really the thing is we're all problem solvers, and that never went away. And so they're just different kinds of problems, different scales of problems. This is why we all are in science. We did a PhD and so on and so forth. So that just continues, and that's I think enduring. And even I would say, if you move away, I mean, things that have to do with more strategic decision making and so on and so forth, there's still problems, and you use the same principles to try and solve them. So that's firmly for me something that I continue to like and love. And of course, as compared to academia, for example, someone mentioned in the beginning that there's a very strong positive feedback to seeing what you do have an impact. and I don't want to get all his thing about impacting lives and so on, but you're really close. I mean, for the most part, you're really close to impacting lives, even if the things that you do fail, and most things fail. But even so, if you understand that those failures are part of, you have to go through those failures to have that final success, you're really in it. And I think that, I mean, it's such a fortunate position to be in that really does drive you. What I do not like, or like less, I should say, is that, and again, this goes back to this idea of working in teams and communicating and so on. We tend to over communicate, and there is a reason for that. And some places over communicate more than others, and it takes a lot of time and effort to do that well. Of course, you get used to it, and so as you do it more and more often, you spend less time, but you do spend a lot of time. So I would say, as compared to academia, where you spend a lot of time teaching and writing grants, in industry, at least in some positions, you spend a lot of time communicating. I don't necessarily dislike it, but sometimes, especially for really matrix organizations like ours, where we're also collaborating with AbbVie, who are geographically isolated, there's a lot of communication, and it needs to be done. And sometimes I feel like I just gave this presentation to somebody else. Now I'll do this again. Okay, fine, I'll just do it. But it's a minor grouse. For the most part, it's good.
Thank you, Sanal. So the next question we have is, what is the one thing that you would change about academic training that would have helped you to be better prepared for your current positions? We can go to Tim.
I think, to be honest, I think I was quite fortunate in my academic training. I think if there's one thing that I could have maybe leveraged a little bit more is, sort of actively seeking out or opening the dialogue to biotech or pharma during my postdoc as well. And I think there are opportunities to collaborate. And, so for example, here at Sanofi, we have money to do external academic collaborations to dive into biological questions and mechanisms that are sort of beyond the scope of what we need to advance a program, but are important to help understand the biology and understand sort of the broader effort, and eventually maybe help, pick patients or find biomarkers, this type of thing. So, I don't have a sort of advice on how to find those opportunities. And I think part of it is through PIs, part of it's through networking, but, I think any kind of exposure during my earlier years to sort of what was going on in industry would have helped, but sometimes that's easier said than done.
Thank you, Tim. So the next question is, how much scope of exploratory research do you have in industry? And if you can compare to academic settings, we would go to Deepa.
I think one of the others might be more appropriate to answer this question, only because they both work in more of a therapeutic area or pharmacology kind of setting. And I work, where I work, we have projects come to us because targets come to us, and we do antibody discovery characterization. However, there is still a lot of scope for, it depends, right? For where I work, I think we have a lot of innovation and we have new technologies that are being developed that we can apply here. So there's definitely a component where we are always looking at what's new, what's out there, and what else you can do to improve or to bring in the latest and greatest and apply it. There's also this part of doing things faster, higher throughput. So taking an assay that's developed somewhere and making it in a different perspective to to apply to the needs here. So I think there's always scope and space to do things that are outside delivering for a project. But it might kind of differ, right? Like sitting here, I don't have the capability of going back and saying, oh, I was interested in this target in cancer research, and I want to work on that. That might not work, but there's definitely room for doing other stuff beyond what you do on what your deliverable or the projects that you're currently working on. It's definitely not as wide as academia. right? It's a different goal. It's a different setup.
Thank you, Deepa. Do you have anything to add, Sanyal, to it?
Sure. I mean, I think it depends on the company. So if you're a very small biotech that's very focused and they have one target, then I think the amount of exploration they're going to be able to do, even in terms of funding, is going to be less. Mid-size and larger companies have a slightly different attitude, especially in the discovery organizations, because they understand completely that discoveries can and do come from everywhere. They're not going to come from entirely within the organization. It's just understood, and they completely get that. So many of these companies have a lot of sponsored research agreements with academic institutions, for example. Calico is unique. I mean, Calico and Biogen, the two companies that I've been at, Biogen had a lot of these. They were more focused because we had focused therapeutic areas. Calico less so, because Calico is focused on aging, so it's not a therapeutic area driven company. So we have a lot of SRAs and a lot of interactions with academic institutions, again, fully understanding that many of these findings will come from elsewhere. So yeah, so you do have, we find that we do have a lot of chance for exploratory work. Now, obviously, exploration not for the sake of exploration, but exploration for, the understanding is that you're going to try to address a certain question that is aligned. So we wouldn't certainly be working on plants, for example, or maybe we will, I don't know. But the thing is that it's focused on, or it's aligned with the overall mission. It depends on the company. Smaller companies, less so. Bigger companies, they fully understand that they need to interact with everybody else.
Yeah, when I joined, so sort of within the neuroscience group here, as Sanyal mentioned, we have sort of focus groups, or we call them clusters, but some focused on sort of neurogenetic research. I'm in the neuroinflammatory and sort of glial biology group. So we're interested in targets that go after those types of mechanisms. And so when I joined the company, I was tasked with finding new targets. And basically given a blank slate. And I think, then you are using all those skills that you've developed before to, understand the literature, understand sort of the robustness of the research that's already published. We had a lot of internal data sets. So from sort of transcriptomic databases, single nucleus databases from patient tissue. We do a lot of sort of deep genetic analysis on these sort of population databases like FinGen. And so I think you have to sort of integrate all of these types of sources and come up with a target. But from the freedom perspective, I think as long as you can make a really strong case for a target and you can sort of replicate a lot of the data in your group and in-house, then there can be a lot of freedom to just go after something that isn't being done in the company.
Thank you, Tim. So we'll quickly go to our live question answer session. We have many comments, and I'm just reading a few of the questions we have, which are new. This is I'm just quoting the question. This is a question especially for the people who have been in multiple places in their careers. How different are the cultures between academia and biotech generally, and how widely have you seen the differences in company cultures between companies? So it's the cultural difference between academia and company.
I guess, I mean, we addressed some of them when we were talking about work-life balance and various things. It depends on what you mean by culture. And it really, I think, draws from, again, so teamwork and so on and so forth. Across companies, yeah, it can vary a lot. I mean, and you'll get a sense, that's partly what the interview is designed to give you a sense for. And you will know. I mean, other than saying that you will know, I don't really have any words of wisdom around this. That's why interviews are important. That is why I think if you think that something might fit, you should interview and you should meet the people there. They do set the tone, they do set the culture. Culture is dependent on often the middle managers. There's top-down culture that's driven, but the middle managers, the ones that you don't hear of, they're the ones that really form the backbone of the company, and they're the ones who hire and retain people. So they determine culture, and those are the folks you meet when you interview. So culture can differ. It's not one simple thing. Biogen is very different from Calico. The two companies that I've been at, I've liked both of them for different reasons, and there are pet peeves that I have for each one of them. If it works for you, you're fine.
I
Thank
would
you.
probably say the same. My two experiences have been Merck and Lilly, both big pharma, and both had good and bad, and they're very different. So I think one of the key things I wanted to add to what Sanil said is an interview, please remember, is a two-way process. So you should go there with as many questions that you need answered to make a decision as much as they ask you questions. So to understand things like this, you should be prepared to ask your interviewer and whoever you meet with questions. So do not go there thinking that it's an examination where they're trying to see if you know the answers to everything. That's not the process. That should not be the goal of the interview. So ask as many questions as you need to. Be prepared for that so that you get the information you need to make the best decision.
Thank you, Deepa. We'll move to the next question. So it says, I don't want you to feel pressure to disclose your salaries, but given that many of us are coming from grad school postdoc, what should we expect in terms of salary benefit entering with a PhD and limited experience? Anyone can answer.
This is again a wide range. It depends. It's very hard to give a range because just from my experience of helping others transition, I've seen ranges because it depends on country, area, state, size of pharma, and what they're funded from, right? I've seen companies pay lesser when they're solely funded by, say an SBIR grant versus, in big pharma, typical salaries also depends on the role and group. So it's very hard to give a number for Glassdoor, and then there are websites online that can help you get a range.
Yeah, I would say from my sort of looking at places like Glassdoor, that'll give you the general idea. And it can range a lot, but that should give you a pretty good sense. I think one of the hardest things for me coming in from the outside was sort of understanding what each title meant and sort of the level and even that changes between companies. Our company just did sort of a company-wide retitling and realignment to sort of a different naming system. So, I think sort of senior scientist is sort of the level you come in after a PhD, typically. The nomenclature can change a little bit, but I think generally that's sort of the idea.
Thank you, Tim. So we almost reach the end. We still have some questions, but I think it would be good if we can get those answered later on from the panelists. And we anyway will be compiling a resource package that we will send to every participant, so we can include the answers in those package. Thank you everyone for joining us today. A special thanks to all the panelists who gave their time and shared their experience. As I mentioned, you will get today's workshop resource package, which will include the unanswered question as well as some other resources that we discussed about. And so keep an eye in your email because we will be emailing to the provided email address that you gave during the registration process. And thank you and have a great day.
Thank you.
Thanks everyone. Good luck.
Thanks. Bye.